Hello, and yes, thank you for the introduction. As was said, my name is Giraud, and today we present part of my PhD thesis about the simulation of water and carbon fluxes in the rhizosphere. So, to start it with, the rhizosphere is the area of soil that is affected biochemically and physically by plant roots. And rhizosphere engineering is when we try to shift the pools and processes that are in the rhizosphere in order to achieve specific aims. So, for instance, in this example, that would be to increase the sequestration of carbon in the rhizosphere. And there is a wide range of management practices that can be done at different scales in order to achieve those aims. And therefore, one useful tool are mechanistic models, because they can help us understand how the management practices affect the rhizosphere and see which management practices might be the most effective. And what we are especially interested in was those multi-domain, multi-scale, multi-process models. So for instance, here you have the representation of a model at the plant and root scale, and with a plant and soil domain, and with a range of processes. And so the advantage of such models is that we can see how the different processes interact across domains and scales and lead to emerging properties. So here's an example from 2010, where we have the same root system and the same root water uptake, but because we have three different soils, we get at the end soil water potential profiles that are completely different. But for my project, we looked at the plant and root scale, the plant and soil domains, and here, so a set of processes, so plant development, microbial dynamic, carbon fluxes, water flow, and with specific soil structure. For the plant scale, we used an FSPM, which are functional structural plant models. With FSPM, we represent explicitly single plants in a 3D space. And they are quite interesting when we want to look at the interactions between gene, environment, and management practices. And for specific outputs such as plant growth, which is then linked with the yield, inner plant fluxes of water and nutrients, plant-plant interaction and competition, and resource and linked to resource acquisition. And so, as mentioned when I gave the title of this presentation, we wanted to look at the effect on plant and soil processes of a dry spell, and that would last for 1 week at different plant development stages. And we had two assumptions that we wanted to test. First was that the drought would lead to higher exudation rates. And second was that these higher exudation rates would lead to higher microbial activity and soil CO2 emission. So that was what we wanted to do. Now I will go in a bit more details about the setup of the models. So starting with our FSPM, we started with cPlantBox, which is an FSPM that has been in development since 2010, which represents a plant using a graph representation, which means our plants are made of a series of nodes, which are points in a 3D space. So they contain their location in a 3D space. And those points are linked by segments. And so this gives us the topology. And the segment stores a set of data that allow us to have information about the morphology of a plant, for instance, the radius of specific segments, the number of xylem vessels, and also the information regarding to the plant functions. So added to this structure, we have module linked to the plant water flow. So we have the loss of water in the leaves via transpiration. This creates a gradient in the plant water potential, and this gradient leads to the flow of water through the plant. and then to the uptake of water in the roots. And we also made the plant growth water dependent. So when the plant water potential is too low, then the growth cannot occur for those specific plant segments. And to represent the carbon fluxes, so in the leaf you have assimilation of carbon, which then also creates a gradient in the solution, and then leads to the flow of a phloem solution through the plant. And then we can have release of exudate and starch in the soils. And we have other sinks and sources in the plants. So for instance, loss of carbon via plant respiration and the storage of carbon as starch, which can then be remobilized. And the plant growth is also dependent on the supply of carbon. So that's for plant. Now I move on to the soil. Where here you see what we use to represent the perirhizal zone. So the perirhizal zone is an area of soil around the roots, which does not have to be of the same volume as the rhizosphere. It's just an area of soil that we select and that we decide to represent, and we have it here with the gray cylinder. And we represent it with a 1D model shown here with this blue line. And so inside this model, we simulate different types of processes and pools. So first we have water, so the flow of water through the 1D models. And then we have different pools of carbon. We have a high molecular weight organic C compounds. So when the plant releases exudates, it is added to, when the plant releases mucilage, it is added to this pool. And we have a low molecular weight organic C compounds. And when the plant releases exudate, it is added to this other pool. And those low molecular weight compounds can be sorbed in the soil, or they can be taken up by the microbes. And the microbes can be either active or dormant. And we divided the microbes in two functional types, oligotrophs and copiotrophs, according to their substrate affinity and metabolic rates. When the microbes die, they go back to the soil organic matter pool, and we also have CO2 emissions, so microbial respiration. So that's our plant and our soil, and they were linked in a global framework. So first we have again our plant model here with the fluxes of water and carbon that are occurring. We have here all of our perirhizal zone models. So you will see for each root segment, we have one specific perirhizal zone model. And so here it's not shown, but then you have a whole ensemble of small 1D models. And we have a 3D soil made of three-dimensional voxels. And so we have two possibilities. Either we are in a voxel that has at least one root segment, like here, and it means that the reactions of the carbon and the reaction of carbon and the interaction with the water are computed by the 1D model. So here we have the different cells. Or we have a voxel that has no roots, and then the different reactions that I talked about earlier are computed in this one voxel at a 3D scale. And that means that we have different resolution in the soil, which allows us to lower the computation time and have a higher resolution in the area of soil that are important to us, which are the area of soil near the root-soil interface. And you can also see actually the 1D model where we have a different resolution. So when we are further away from the root surface, we have higher cell, so a lower resolution. And when we go nearer to the root surface, we have those smaller cells, and so a higher resolution. So that is our model. And then we did a first calibration for a generic C3 monocot and for different microbial parameter sets, although today we only present the result for one specific microbial parameter set. For calibration, we use experimental results and literature data. And we had three scenarios. So we have first our baseline scenario where we simulated empirically the plant growth between day 1 and 10. And then we started with all of our functional models. So for instance, the plant growth dependent on the water and carbon supply. And in the baseline scenario, the soil water potential was always high and constant. And then we had our early dry spell, which is like the baseline. But at the beginning of day 11, we simulated a sharp drop in the soil water content. And then the soil water content became dependent on the plant water uptake. So as the plant loses water and takes water from the soil becomes drier. And then at day 18, we go back to the default soil water content. So that creates a small wetting drying cycle. And for the late dry spell, the same thing happens, but 1 week later. And so we wanted to look at the effect of those different scenarios for one specific plant and microbial parameter set regarding the allocation of carbon. And so now we move on to our results, where here you see the time series between the start of the implementation of the functional models, so from day 10 to 25. So the first vertical dotted line that you see here is the start of the early dry spell. The second dotted line is the end of the early dry spell and the start of the late dry spell. And the third dotted line is the end of the late dry spell. And the color of the lines give the different scenarios. So in black, you have the baseline. In green, you have the plant that was in the early dry spell scenario, and in orange, the plant that was in the late dry spell scenario. So the first panel here shows the mean plant water potential. And so as expected, during the dry spell, we have a drop in plant water potential. And also you see the day-night cycle. So during the day, the water potential drops, and at night, the stomata closes and the plant water potential increases again. And we see some slight difference between the two scenarios in the mean plant water potential. So for instance, at the start of the early dry spell, we see a sharper drop in the plant water potential compared with the late dry spell, and that's linked to the size of the root system. So for the late dry spell, because we have more root surface area, the plant is better able to withstand the drought. But with the late dry spell, because you have this higher transpiration, because the root system is bigger and the leaf surface is bigger, by the end of the simulation, actually we get the lowest plant water potential with the late dry spell. Whereas with at the end of the early drought, it is a little bit higher. On the second panel, we see the cumulative amount of carbon that is used for growth during the simulation. And then we can see the effect that the water potential has on the growth. So during the dry spell, because the water potential of the plant drops, the plant growth almost completely stops in the early dry spell. And for the last days of the late dry spell, we have no growth at all. And in panel C, we then see the resulting plant exudation, where we have actually diverging results between the early dry spell and the late dry spell. So that is because in the early dry spell, the plant is not quite so grown yet. So the leaf area is still somewhat smaller, the root surface area is still a bit small. And because the plant almost does not grow, then it means that the plant capacity to release exudate is much smaller. And so we get a smaller cumulative exudation. But with the late dry spell, by the time it starts, the plant is already somewhat bigger, with already more starch that is stored, a higher leaf surface area and root surface area. So it has already much more carbon. And the fact that less carbon is used for growth, so we have one less carbon sink, means that we have more carbon that is available for exudation. And so we get a higher cumulative exudation. So that was for the plant. And now we talk about the last three panels, which are linked to the results in the 1D models I talked about earlier, where we have all of those different cells. And here we have 1 point for each cell of each 1D model. So as I said also before, we have one perirhizal zone model for each root segment, And so here it's all of rows of our pay-horizontal models of all of our root segments for one root system. And they are ordered along the x-axis according to their distance from the root surface. So it means that here it's all the points that are directly at the root cell interface, and here we are further away from the roots. So what we can first see is that we have a day-night balance. So during the day, we have an exudation. So the amount of low-weight organic C compounds increase. And also because you have this uptake of water, the water content in the soil decreases. And so that also leads to higher concentrations because all those compounds are in the water phase. And at night, because the carbon are eaten up by the microbes, the concentration go down again and are then even smaller than further away from the root. And we see the effect that this has on the microbial activity, which is represented by the coloration of the points. So when you have high colorations, when you have high concentrations, the points become yellow, and that means that the microbes are more active. And at night, when they have taken up all of the carbon available, they become dormant again. So that is what we call a starving survival lifestyle, where the microbes wake up as long as there is a pulse of resource, and then they become dormant again once they have depleted it. When you see one of the square becoming red, it means that it is during the dry spell. And we see that here we have a much smaller balance. So the concentration remains high during most of the day-night cycle. And that is because as we do not have the usage of carbon for growth, the exudation can be maintained during the whole cycle. And because we have even less water content during the spells, we reach even higher concentrations. In spite of this, you see that the points turn blue and green, and that is because the microbes suffer water stress, and because of this water stress, they become partly dormant again. And now we move on to our results, but at the 3D bulk soil scale. And here we wanted to look at the allocation of carbon between the different scenarios. So as before, you have in green the early dry spell scenario and in orange the late dry spell scenario. And we looked at the amount of carbon in different pools compared with the baseline. So when you have a line at 0, it means that you have the exact same amounts compared with the baseline. And so for the late dry spell, we are at 0 until the dry spell starts, so at day 18. And that makes sense because before then we have the exact same condition between the baseline and the late dry spell scenario. So starting with the carbon that is in the solute pool, during the dry spell, this amount increases, even though, for instance, in the early dry spell, we saw earlier that the cumulative carbon exudated was lower compared with the baseline. And why does it happen? So it's an effect of the root system size. So during the dry spell, the root system increases not as quickly. And so it means that the volume of soil in which the plant releases carbon is smaller. And so it's harder for the microbe to access the carbon that is being exudated, and then it tends to accumulate more instead of going into the microbial pool. And so, for instance, we see then for the early dry spell that we go back, also with the late dry spell, we go back to amounts that are similar to the baseline by the end of the simulation. And now we look at the amount of activated microbes, and we see an effect that resembles the Birch effect. So the Birch effect is the idea that when we have wetting drying cycles, during the dry spell, the resources accumulate in the soil because microbes cannot take them up because they suffer from stress. And when we have a wet pulse, then the microbes become activated again and they can profit from the solute that had accumulated. And then we get a small activation pulse and then a small CO2 emission pulse. And that is a little bit what we see with our early dry spell, where the activation of microbes goes down and then goes up with the pulse. And we see also the CO2 emission that goes a little bit up after the wet pulse. But we only call it a Birch-like effect because we don't represent all the processes that are involved in this phenomenon. And then we can look at the total amount of microbes and emitted CO2. So first, for the total amount of microbes, we have divergent results between the early and late dry spell, and that is linked to the total amount of carbon that is emitted. So because you have those very active microbes that can really take up what is released by the plant with their starving survival lifestyle, the total amount of microbes at the end of the simulation is really dependent on how much the plant exudated. So the plant exudated more in the late dry spell, so we have more microbes, and exudated less in the late dry spell, so we have less microbes. And we see a strong link between emitted microbial CO2 and total microbial biomass. So there's a stronger correlation than with activated microbes. And that's because even though the dormant microbes do less respiration, because the vast majority of microbes are dormant, we have these stronger links. And so now we go back to our assumptions. So the first one being that droughts will lead to higher exudation rates. So for us, that was dependent on the development stage of a plant. And regarding the idea that it will lead to a higher development of a microbial community and CO2 emission, that was dependent on whether we did have these high exudation rates. And even though I do not show it here, it was also dependent on the calibration of our microbial community. So when we have this very active microbial community, they could profit from the additional carbon. But for other simulation that we conducted with less active microbial community, with, for instance, lower maximum carbon uptake rates, then the microbial community suffered more from water stress than they profited from the additional carbon. And so those were for our two assumptions. And we also compared our results with a meta-analysis from Deng et al. From 2021, where they looked at the effect of droughts on the allocation of carbon. So like they did, we saw a lower shoot and root biomass because the plant was under water stress. And like they did also, we had higher concentration of dissolved organic carbon during the drought, partly because we had sometimes higher plant carbon releases, but mainly because the saltwater content was lower. In some cases, we had a higher amount of microbes, so with that we have with the green arrow. But as I said before, with some of our calibration, we have a lower development of the microbial biomass. So that was dependent on our calibration. Do we have those more active microbes or not? And also the age of the plant when the drought started. And for microbial released CO2, so it varied like the microbial biomass. So we did not always have the same results as they observed in their meta-analysis. And for the total soil organic carbon, so we generally always observed an increase, contrary to what Deng et al. Observed. And that is probably in part linked to the fact that we did not represent the addition of carbon from the plant via plant residues. So normally less plant biomass would mean that we will have less dead root, less dead leaf, and that would decrease the soil carbon. But as we did not include this process, we did not see these relative differences. And also for us, we simulated a smaller time span as in the meta-analysis of Deng et al. So yes, using our model, we could compare with data from the literature and better see what are processes that are still missing. And now we move on to the overall conclusion. So we saw different strategies by the plant and the microbes according to the calibration that we used. So different strategy in how they responded to different stresses. And the carbon allocation was a very emergent and plastic output of the model that was dependent on the interactions across scales and domains. And so this model that I show, so it includes a lot of different processes, but we aim to make it flexible enough so that potential user can decide if they want only to look at plant structure or also include specific processes of a plant and also of the soil. And they can use it to look at the carbon allocation under various and different limiting conditions that interact, and to look also at phenotype performance and management measures. And so right now I discussed more trends in the model output than really precise numerical results. And that because we had this first rough calibration, but now we are inside the project crop 2, which is the second phase of my project. And we want to do, we are doing field experiment and laboratory experiment to do more precise calibration and test of the model. And also here you see a photography of a field experiment from last year, where we looked at the development of wheat plant under different nitrogen fertilization levels. And that is because we also want to include nitrogen balance in the model. So that is also a future. So the next steps that are being done. And here you see all the other people that are involved also in the crop project on both the experimental and the simulation side that have also allowed for the results I've presented today. And as last acknowledgement, so cPlanbox, as I say, is a model that has been developed for a long time. And so here you see a list of current and former contributors. And so, yes, that have also then led to the output that I have shown today. And yes, and we have a lot of different studies if you're interested to learn more about cPlanbox. So that have been published so in the last years, for instance, the interaction with light, with phosphorus supply, and so on. And so yes, I come now to the end of my presentation. Do not hesitate if you have questions.